हो गए? मुझे दिख नहीं रहा है मेरा यूट्यूब पे। ठीक है। Hello, good evening. Good evening, everyone. Am I audible? My voice is clear. Good evening to all of you. I hope my voice is clear to all of you. So, so the result of NIPER will be declared soon. So, do not worry. I think within 3-4 days, your result will be out. So, do not worry. It will be out soon. So last class we are talking about carbohydrate. In this today's class, good evening to all of you. I hope I'm audible and visible clearly to all of you. So last class we are talking about this uh, carbohydrates. So in this video or in this live class, we are going to discuss about biotechnology. So what is biotechnology? First understand. So when you heard this word biotechnology, so what is biotechnology basically? So biotechnology, if we break this word, one will be bio, another one will be technology. So the technology or the techniques that is associated with the living system or living organism, we call it as a biology, a biotechnology. So this biotechnology field maybe deals with the protein, deals with DNA, RNA, many things. Either we talked about their expression or their separation or their recombination. Okay, so the topic that today which we are going to discuss today is your PCR. Okay, today what we are going to discuss is PCR. That is polymerase chain reaction. Let's understand its experiment from this one. So why it is name is polymerase? Because you are using a enzyme name is DNA polymerase. DNA polymerase or specific if I will specify that is your tag DNA polymerase. Okay, this is more specific. And why it is chain reaction? Because this polymerization reaction will be carried out for certain time period of time. Okay, so that's why we called it as a chain reaction. Okay, so let's start. So this let me discuss you first overally. So today what we are going to discuss is we are going to discuss on this PCR. Okay, so PCR stands for your polymerase chain reaction. It starts for polymerase chain reaction. This polymerase chain reaction, first time it was discovered or developed by American scientist that is name is Gary Mullis. Many times this question I've asked. Okay. So what is this PCR basically? PCR basically a cell-free amplification system. It is basically a cell-free amplification system. So what is cell-free amplification system? So basically amplification, matlab formation of one or we can call uh, copy formation of copies of DNA or RNA. So these amplification, basically this formation of this one copy, or just it is just like PCR is just like a Xerox machine. So if I simply tell, PCR is just like a Xerox machine. So like in Xerox machine, we will place a paper and we wanted 10 copies, 12 copies, or 15 copies. So we place that paper and we type that yes, we need 15 copies. So that 15 copies will be exactly what that we give the command, same type, exactly same copies will be generated. Or PCR is just like the same. Okay, so amplification occurs. Why we call it as a cell free? Because this amplification system is occurring in vitro, because it is carried out outside the cell. Okay, that is your cell free amplification system. So this basically, if we talk about its steps, it has three major steps. Three major steps. First step is denaturation. So denaturation means breakdown. So what will break down? I will discuss. Next step is annealing. So what is annealing? Annealing means joining. And third important step is synthesis or or extension. Synthesis or extension. There are three major steps. One is denaturation, second one is annealing, third one is synthesis or extension. So what is this denaturation? Basically, denaturation means breakdown of these two strands. So I'm just explaining the things. Try to understand first, then we will enter to the content. Okay. So whenever you feel any doubt, just put your question inside on the chat box. So first is, let's give you an example. See, this is one strand. This is a DNA. Okay. So first step, what I discussed as a denaturation. So what happens in denaturation? Two strands will be separated from each other. What happens? These two strands will separated from each other. Okay. So you know one strand is from 3 prime to 5 prime. Another one is from 5 prime to, one is from 3 prime to 5 prime direction. Another one is from 5 prime to 3 prime direction. So once denaturation, or when these two strands basically separated from each other, next step is annealing or annealing step. Next step is annealing. So what happens in annealing? So let me draw this first. This is the two strands of DNA. One is from 3 prime to 5 prime. Another one is from 5 prime to 3 prime. Okay. So here now primers will be bind. Okay. Now what happens? Now primers will be bind. So there are two different type of primers are used. One is called forward primer, another one is called reverse primer. Okay. So one primer is called forward. This is your reverse. Now after this, the final step is forward primer. It will move from this direction. Reverse primer will move from this direction. Okay. And last important step is extension. Last important step is extension or synthesis. Extension or synthesis. So now two complete DNA strands will be formed. Now from this single strand, two copies of DNA will be formed. This is your mechanism of PCR. So first tell me you understood or not. Tell me in the chat box. First you understood or not. So primers are short nucleotide sequence. So what is primer? Primers are, these are short polynucleotide sequence. These primer are basically of two types. Okay, one is called FP or forward primer, another one is RP or reverse primer. So primers are not. If I will give a simple example, for suppose you want to check that in that particular species inflammation is occur or not. Okay, if you have to check inflammation of is occur. For inflammation, you have to check the level of NF kappa B, nuclear factor kappa erythroid beta factor. So this NF kappa level has to be checked in this sample. So for this, you have to purchase two primer. One is your forward NF kappa beta primer. Another one is your reverse NF kappa beta pi. One is forward NF kappa beta primer. Another one is your reverse NF kappa beta. Okay, so this NF kappa beta primer, when you add to the sample, if it will bind and copy, multiple copies are formed, that means that particular sample has NF kappa beta positive. That means that that particular person or that particular individual is positive for inflammation. So this is actually the primers is looked at. Okay, now we will talk to the content first. So the polymerase chain reaction or PCR, this is a laboratory technique for generating large quantities of specified DNA. It is used for generating large quantity of specified DNA. PCR is nothing but this is a cell-free amplification technique. What is it? It is a cell-free amplification technique for the synthesis of identical copies of any data of interest. That means you are just doing the copy synthesis. Okay. So for this technique, he got Nobel Prize in the year 1993. Please remember. So what happened basically, the principle that we have discussed now, the double stranded DNA of interest basically denatured. Denatured means breakdown or separated into two individual strands. Now this each strand then allowed to hybridize with a primer called renaturation or annealing, and the final step is synthesis of DNA template or called as extension or synthesis. So this extension or synthesis step basically required which enzyme? You required DNA polymerase enzyme. It required DNA polymerase enzyme. Our specific is TAC DNA polymerase. Thermus aquaticus, we will discuss one by one in detail. Okay, so question can be asked. So this, it takes three different steps. One is your denaturation, second one is annealing, the third one is your renaturation or synthesis. Okay. Next is, let's talk about the steps of this PCR. So what happens in PCR is, so first step is your denaturation. We have discussed. So denaturation means we are breaking. For breaking, what we need? We need a high temperature. Here temperature around 95 degrees. Temperature around 92 to 95 degrees centigrade. Okay, because denaturation step is basically, so we are applying heat. So due to this application of heat, so what happens here? The two strands basically separated from each other due to application of the heat. So once you applied the heat, next step what you will do? You will go for cooling. Okay, so because you know now this, unless until the temperature has not reduced to for a certain temperature, this primer cannot bind. If I will give a simple example. For suppose uh, when suppose two friends, so two friends fight between each other. Okay, that these two students started fighting between each other. One friend has uh, became more angry, and we told her like that particular individual having high temperature in the brain. Your brain has high temperature. So it's just like a science. Okay, so when the person is or the temperature is too high, that primer cannot bind to this separated strand. For the binding of primer to the separate strand, the temperature has to be reduced from 92 to 95 degree to around 55 degrees centigrade. So here the temperature will be around 55 degrees centigrade. Okay, so the temperature is around 55 degrees centigrade. So this step is called heating. This step is called heating. This step is cooling, and the final step is again heating. So now next, my your question is, sir, sir, we have reduced the temperature for binding. Now why we are increasing the temperature? First, you told that first we need to increase the temperature so that DNA strands get separated from each other. So we did that. Next step, what you told, sir, you told that you have to reduce the temperature to 55 degree temperature so that this primer will go and uh, binds to this separated strand. Now you are telling that again you have to do heating because again you have to take the temperature to around 72 degree. Again, this temperature has to be reached 72 degree because 72 degree is a optimum temperature for the enzyme DNA polymerase. So this 72 degree is the optimum temperature for this is the optimum temperature for this enzyme. Okay, so this step is called heating step. So these three steps are basically the major steps of PCR. So first step of PCR is denaturation. So on by rising the temperature to around 95 degrees centigrade about 1 minute, the DNA gets denatured and the two strands get separated. Second step is renaturation or annealing. In this step, so primer are start pairing with the complementary region of the target DNA. The template DNA should be lower to 50 to 70. So it is the exact temperature is 55 degree. Please remember 55 degree. And the annealing will be start typically below 5 degree of the calculated TM. TM stands for melting temperature. One of the important thing that you have to remember here is the primer. The important thing is the primer will bind the primer will bind to 3 prime OH. And you have to remember primers will bind 3 prime dash of this DNA doubles. Okay, it has to remember question can be asked. Okay, so this is last step is DNA synthesis or primer extension. So the initiation of DNA synthesis occurs at 3 prime hydroxyl end of each primer. The primers are extended by joining to the base complementary to the DNA strand. Okay, these primers are extended by joining to this complementary DNA strand. The synthesis process of PCR is quite comparable and DNA replication of the leading strand. So here the enzyme that play a key role is your tag DNA polymerase. The enzyme plays a key role over here is tag DNA polymerase, and the optimum temperature for this enzyme is 75 degrees. Okay, so please note it down. Then I will discuss some important other points. Please write it down. Please write it down. Please write it down. First, tell me in the chat box, understood or not? First, tell me in the chat box, understood or not? First, tell me in the chat box, understood or not? Okay. Now let's summarize by this reading this table. So first step is denaturation. Here temperature will be around 95 degree. This question was asked in GPAT 2020. And this will start for this temp duration will be given for 1 minute, and here heating will be there. Next is aniline, where temperature is around 55 degree. So the duration of this exposure is 45 seconds, and this is basically a cooling step. Final step is synthesis. The temperature will be around 75 degrees centigrade. The time will be 3 to 5 minutes. You have to give the exposure. Okay, you did not understood about primer. Okay, let me tell you once again. So primers. So why primers? So primers are basically a particular protein or particular what can call particular uh, what we can call particular gene that has to bind to that particular your DNA stand for the simple. I'm giving you an example. For suppose you are having you want to check the for suppose you are a researcher. You want to check that in particular tissue. For suppose I am working on cancer. So for suppose I am checking in cancer, liver cancer, what is the expression of P53 gene. Okay, what I am telling. So we are checking about P53 gene in liver cancer. So what I will do, I will add this two primer of P53. So one primer of P53 is your forward primer, one primer of 53 is reverse primer. So that means, so same P53, because na DNA stands are, so why we add this forward primer and reverse primer is there is a logic, because the two strands of DNA are anti-parallel to each other. Yes or no? Anti-parallel to each other. So what does meaning of this anti-parallel? Anti-parallel means one strand will bind from 5 prime to 3 prime direction. Another one is 3 prime to 5 prime direction. Okay. So the as the anti-parallel in nature. So, and the binding of the primer will be always at 3 prime end. So it will always bind to this 3 prime end. If both are forward primer, then one will be moves in this direction, another will be moving opposite direction. Okay. So for the due to this anti-parallel nature of DNA strands, so these forward primer and reverse primers have used. Sir, in denaturation and annealing, no enzyme required. Yes. No. Here no, because the synthesis has not started. In those two steps, in denaturation and annealing, this is the separation phase. So actual synthesis is takes in this step. So for in this step exactly, you need this enzyme. Okay, in this step particularly, you need this enzyme. Okay, now you understood what is primer and what is this, how this primer shall work. I will explain you what are the things we need in PCR. So what are the component or what are the essential component of the PCR? So if you want to get important e-notes for B Farm and other GPAT and other pharma competitive examination, you need to join this batch in the GDC classes app. So you have to download the GDC classes app first, then go to the batch section. you have to go to click this plus icon. Then you have to put this code GDC E26. Then you will add it to this batch. And once you add it to this batch, you will uh, get all the input and E nodes over there. So again, I am discussing this mechanism. Please understood very carefully. So first step is denaturation. Denaturation means here your two DNA strands are separated. So that is called as template strand. What it is called? It's called, this is your parent strand. From the parent strand, you get two template strands. Now, after getting these two templates done, then next step is aniline. Okay. So aniline you have to add. So in this aniline, so here no, actually in this step you have to add, but the actual function of this DNA polymerase will be start in this step. Okay. So you have to add this DNA polymerase. So you have to add this nucleotide, that is DTTP, DCTP, D. We shortly call it as a DNTPs. Shortly, we call it as a DNTPs. We contains TTP, CTP, ATP, GTP because DNA contains nitrogenous bases. These nitrogenous bases are two types. Yes or no? One is your purine, another one is your pyrimidine. So what are the purine? Adenine and guanine are the purine. Thiamine and cytosine are the pyrimidine present in DNA. That's why we add this DNTP. After aniline step, now this primer will be bind. So now this primer will be bind. One is your forward primer, another one is your reverse primer. Once this bind to this primer, this DNA polymerase, this is your tag DNA polymerase. This is your tag DNA polymerase. Now this tag DNA polymerase will be binds. So once it binds, it will synthesize a completely new strand. So this is the mechanism. Okay. This is the exact mechanism where how this technique basically work. Okay. Understood. Now, next thing is what we require. What are the components requirements? So what are the requirements? So first, what we require? Yes, first most important thing, the enzyme, tag DNA polymerase. Tag DNA polymerase. Second point, DNTP, which consisting of DATP, DGTP, DTP, DTTP. Third is your buffers. Okay. Third is your buffer. Next is fourth one is primers. One is your forward primer and second one is your reverse primer. Fifth one is cDNA. I will explain what is cDNA. Sixth one is, yes, magnesium required. Magnesium as a cofactor, it is required. Okay. These are the basic component of this our PCR. Okay. So thermostable DNA polymerase or tag DNA polymerase, which can stand up to 95-degree. Primers, both forward primer and reverse primer. DNTPs like ATP, ZTP, CTP, and TTP. Divalent cation like magnesium as a cofactor for DNA polymerase. Buffers and template DNA stuff. Okay. So let's see. So this is the typical PCR machine. And this is your, on the white crystal you have seen over here, this is your RNA. So I am giving you a simple example how we do PCR in our lab. So for suppose I want to check, so let's take that same example. I want to check that P53 expression in liver cancer. I want to check this P53 expression in liver cancer. So what I will do? So what we will do first, we will take that liver. We will take that liver. From the liver, we have to isolate this RNA and exactly the mRNA. So we have to isolate this mRNA. Now, this mRNA or RNA is unstable. It is unstable. It is unstable because it is easily degradable. Because it is easily degradable if change in temperature occur. If minor change in temperature seen. Okay. So what we will do, we will synthesize a complementary DNA from the, we will synthesize the cDNA. cDNA is nothing but this is just a complementary, it is just complementary to this mRNA. It is just complementary to this mRNA. It is complementary to this mRNA. Now this cDNA that is synthesized over here is highly stable, can be stored and can be stored for, okay. Now, once you synthesize this cDNA, okay, you have to add this other component. One, you have to take a PCR tubes, okay. This is called a PCR tube. So to this PCR tube, you have to add this cDNA and your DNTPs, your primers, your magnesium, everything you have to add. And in this PCR tube, you have, once you add this, mix this all component, you have to place inside this instrument. In this instrument, there is a software where you can set the temperature. Where in this you can set the temperature. Here you can set the temperature according to our step, that is 92, 55, and 70. So that temperature you have to set in this machine. Once you set this temperature, yes, machine will now understood that yes, we have to perform this thing. So once this complete, you will get a multiple copies of DNA. What you will get? You will get a multiple copies of DNA. Okay. So now let's talk about this DNA polymerase. So when this DNA polymerase initially developed by this uh, scientist, this 𝘂𝙎𝙎𝙎𝙎𝙬, that initial fragments are called as neon fragments. Okay, neon fragments initial. So what is the disadvantages of this? Is this is hormolabile. So initial the fragment which were developed by 𝘂𝙎𝙎𝙎𝙬 is called as neon fragment. So these fragments are hormolabile. What are they? They are hormolabile. Okay, so these are called as hormolabile compound because any change in temperature, because here the temperature varies from up temperature rise up to 92 degrees centigrade. So the major advantages of this Leon fragment is you have to add fresh DNA polymerase after each cycle. You have to add fresh DNA polymerase after each cycle. So to avoid this or to overcome this problem, so this stacked DNA polymerase is developed. So in this original PCR, the neon fragments are introduced from the E. coli are used. The enzyme gets denatured at higher temperature. Therefore, fresh enzyme need to be added to this each site. These neon fragments are what are they? These are basically large protein fragment of DNA polymerase 1 from the E. coli. So when a breakthrough, that is now this is becoming a bigger problem. So what? Template DNA. Okay, let me complete this. I will explain this template DNA. Now, when this problem has arisen, now this modification has done. So what this modification? In this modification, this DNA polymerase now replaced with a heat thermostable enzyme called as Taq DNA polymerase. This DNA polymerase is replaced with a thermostable enzyme called Taq DNA polymerase. And this Taq DNA polymerase can resist the temperature of to 92 to 95 degrees centigrade. It can bear the temperature or it can stable at the temperature around 92 to 95 degrees centigrade, although its optimum temperature is at 94 degrees. Okay, it can register temperature up to 94 degree and its optimum temperature is 70 to 80 degrees. That's why we take it at the 72 as the ideal temperature. This tag DNA polymerase is isolated from a species called Hormus aquaticus and it is thermostable. Many times from this part, 3 to 4 times in GPAT question has been asked. Okay, so tell me, understood or not? First, tell me, understood or not? Yes, tell me, understood or not? If you understood, give a thumbs up in the chat box. Please show some excitation in this chat box. It is looking like a dull session is going on. Please show some excitation in the chat box. Okay. So now one question has asked in the chat box that what is template stand? Okay. Let me explain what is this template stand. So when you know the first step, so first step of this PCR technique is denaturation. So parent stand means when two stands are joined together by uh, two stands are joined together. This stand is basically called as parent stand. When two stands are joined with each other, two stands are joined with each other. When we applied the temperature around 95 degree, now the stands became break. Okay, now this DNA stand became break and it is called as template stand. Template stand is nothing but a single strand of DNA. Okay, so template stand is nothing but it is a what? It is a single strand of DNA. Okay. Now this single strand, when this primer is bind to this single strand, it will synthesize a what? It will synthesize the its complementary strand. So this when this synthesize this new strand is called as complementary strand. This is called as complementary. Okay, clear? Now, so let's see its uh, application. So PCR is used for the parental diagnosis of inherited disease like your sickle cell anemia, beta thalassemia, phenylketonuria for the detection by PCR. Diagnosis of retroviral infection like PCR, which physically forms the cDNA. It is used for the diagnosis of retroviral infection like your HIV infection. Diagnosis of bacterial disease like your Mycobacterium tuberculosis. Diagnosis of cancer. Determination of sex of embryos. Okay. Sex of humans and livestock embryos in fertilized in vitro can also be determined by this PCR technique. Okay. Other than that, it is also used for the genome sequencing. It is also used for genome sequencing. He also used in case of evolutionary biology, also used in forensic medicine for the identification of criminals. Okay, now you understood how many cycles are required to produce complete DNA. See, in one cycle, okay, in one cycle, you will after completion of one cycle, you will form from one DNA, two strands will be formed. See how this is one DNA. After one cycle, two copies are formed. In next cycle, four copies are formed. In next cycle, 16 copies are formed. So like this, your cycle is basically going on. In one cycle, so one cycle means what? You know, na? So one cycle means denaturation plus aniline plus synthesis. When these three step completes, that is called one cycle. Okay? When these three step completes, that is called one complete cycle. Okay? Now understood or not? Tell me. When these three steps complete, that is called one cycle of PCR is complete. Okay? Understood? Understood or not? Now we will discuss some MCQ. Okay, before discussing that MCQ, so there is a small announcement. So that there is a big announcement by GDC that their new batch has been launched for GPAT 2026 and 2027 in specially English language. Okay, so this is consisting of both 2 year and single year program. For the single year program, the cost is 14,000. For 2 year program, the cost is 18,000. Okay, what you will get this batch? You will get 500 plus live classes, hard copy books, previous year questions, daily practice paper, online test series with detailed explanation. Okay, this course is highly recommended by the GPAT toppers. You can ask any of them. So they will purely 100% recommended these prime batch courses. In this prime batch, we basically teach all of the students from the very grassroots level. From the very grassroots level to the advanced level journey is basically we train our students in such a way that particular students can understand the from the basics to can have the capacity to solve the basic to higher level of questions. So for the joining, you can contact in this number and you share this information with your friends. Okay, so we are going to start our new batch from 29th of June. Okay, from this month, so we are going to start this new batch. Let's discuss some MCQs. Okay, yes. If they ask how many DNA from Acha, you ask about calculation. Okay, we will explain. No issue. Just give me a second. So how you are going to do this calculation? You can ground this, you can done this calculation by a simple formula. That formula is, what is that formula? NF is equal to Ni into 2 to the power n. This is the formula. NF is your final number, final number of DNA. Okay. So Ni means initial number. How many initial number of DNA you have? Because before uh, keeping the when we add the solution, we do the all the calculation. So Ni stands for this initial number. So initially, how much quantity of DNA you have given, and there is a what we can call, there is a uh, option where we can place how many cycle we want to run. So usually run from 20 to 30 cycle. Usually we run for 20 to 30 cycles. So if we put all this information, you will get this final number. So NF, that is final number DNA, is equal to initial number of DNA into 2 to the power n. N stands for number of cycles. Okay. Actually, I seen a question: how many will be synthesized after 10 cycle of PCR? Yes, like this. This is the formula. So see, after 20 cycle, the see, after 20 cycles, 1 into 10 to the power 6, or we can call 1 lakh DNA can be synthesized. Okay, one. So similarly, if we 10 cycle, it will be reduced to half. So after 20 cycle, 1 into 10 to the power 6 total DNA can be synthesized. If question will be asked about 10 cycle, it is not, it is just of this half, 1 into 10 to the power 6 divided by 2. Okay, now understood. Now tell me, you understood or not? Yes, reply me in the chat box. Now you understood or not? Okay. If understood, then just give me a thumbs up in the chat box. Show some excitation in the group. Okay, it is looking like a very dull history class. So it is not like a history class. It is just like a interesting. We are just learning a technique. Okay. So show some excitation in the chat box. Okay. So let's start the MCQ. So the first question is, during denaturation cycle in PCR. So first question is, during denaturation, so the PCR, the temperature rise to 90 to 98 degrees centigrade, 60 to 68 degrees centigrade, 70 to 78, yeah, 80 to 80. Yes, answer this question. Students, answer this question. Very good. Option A is the correct answer. Very good. Option A is the correct answer. Very good. Next question. DNA amplification by PCR used. Thermus aquaticus DNA polymerase, DNA topoisomerase, RNA polymerase, या DNA helicase. RNA helicase. Answer this question number 2. DNA amplification by PCR used 𝘎𝑃𝑎 aquaticus DNA polymerase. Okay, very good. Option A is the correct answer. Next question. Which of the following enzyme is isolated from 𝘎aquaticus? This question was asked in 2015. DNA for tag polymerase, tag ligase, tag kinase, or tag nuclease. The formula again I am writing NF is equal to Ni into 2 to the power n. 2 to the power n. This is the formula. I write that after 20 cycle, okay, after 20 cycle, if question will be asked about how many DNA will be, copies of DNA will be formed, the amount is 1 into 10 to the power 6. This much amount will be synthesized. Now it is clear. Now it is clear is not. This is the formula I talked about. The after 20 cycle, how many DNA will be synthesized. Tell me first, it is clear or not? Clear or not? Tell me first. Next question. The PCR technique was developed by Kohler, Altman, Milstein, या Karimulis? Answer this question. The PCR technique was developed by Kohler, Altman, Milstein, या Karimulis? Yes, answer this question. Who developed this PCR technique? Very good. Karimulis. Karimulis is the correct answer. Next question. The polymerase chain reaction, it is a DNA sequencing technique, it is a DNA degradation technique, it is a DNA amplification technique, all of the above. PCR technique was, the next question is, the polymerase chain reaction is a DNA sequencing technique, DNA degradation technique, DNA amplification technique, all of the above. Very good. DNA amplification technique. Option C is the correct answer. Next question. Which of the following is the basic requirement of a PCR reaction? Two oligonucleotide primers, DNA segmented segments to be amplified, a heat stable DNA primer. Yes, two oligonucleotide primer, that means your forward primer and reverse primer. One is DNA segment to be amplified. Yes, the cDNA, a heat stable DNA polymerase, that is your tag DNA polymerase. Yes, we require all this. All the things will be required. Okay. Next question. Which of the following is the first and most important steps in the polymerase chain reaction? First and most important step. Okay. First and most important. Annealing, primer extension, denaturation, yeah, none of the above. Answer this question. Very good. See the clear answer. Answer next question. Very good. Denaturation. Denaturation step. So denaturation is the most important step in polymerase chain reaction. Next question. Polymerase used for PCR is extracted from. Polymerase used in PCR extracted from Homo sapiens, 𝘎𝑃𝑎 aquaticus, Escherichia coli, या Saccharomyces cerevisiae. Thermus aquaticus. Option B. I told you na. Tag DNA polymerase is isolated from Thermus aquaticus. Option B is the correct answer. Next question. Okay. So this was the last question. Okay. Dear students, do you have any question? Dear students, do you have any questions to ask? Very good. Option B is the correct answer. Any questions to ask? Any doubt? Please tell me this session was helpful for you or not? First tell me. The session that we covered today, it was helpful for you or not? First tell me. First tell me the session we covered today was helpful for you guys or not? You need such type of more session or not? Tell me. Please give a positive comment after this class. In this video, please leave a positive comment so that we will bring more such contents for you guys. Okay? Please put some positive comments after this session in this video. Okay? So that's all for today from my side. Thank you. Thank you so much for joining the session. In the next class, we will bring with a new concept for you guys. Thank you. Thank you so much. Thank you. Thank you so much.